Thank you for coming. So, the purpose of today is just to kind of introduce, those of us here at the University of Georgia to the BioFoundry. One of the things I probably should have told you, the thing, we probably would have even more people here, is the BioFoundry, the way it's built, is we have $200,000 a year for collaborations to facilitate your research, right? For us to use our expertise of the team to help facilitate your research. And all you basically have to do is write a little 1 or 2 page grant proposal, right? You basically sit down with us, write this little proposal, and we'll do the project for you, or we'll train your people how to do the project on some of our cutting edge technology, right? And so with that, I just want to say welcome, thanks for coming. So this is about a grant that we received 6 months ago now. Wow, that's time flies. From the National Science Foundation for 6 years. It's a $18 million proposal, and it's all focused on glycoscience. And so my name is Lance Wells. I'm a professor here at the CCRC and also a professor of biochemistry and molecular biology. And I'm the PI on the proposal. I'm going to give you just a brief introduction of what the BioFoundry is trying to accomplish. And I'll talk a little bit about some of our glycoprotein MS resources, and then I'll hand it over to some of the other co-Is who will talk about various things. And then importantly, we want to open it up and talk to you and have you ask us questions about how we can work together, right? Because the whole idea of this BioFoundry is not to provide money for my lab and Kelly's lab and Paris' lab, right? That's a good thing, but it's to spread the love, right? And get glycobiology out to the community. And so this is the team. Paris Azadi, Aaron Dolan, Kelly Mormon Bree, and Cannon. You're going to hear from almost everybody except for Bree. And then we also have a lot of senior personnel who kind of round out our rough edges, have expertises that we don't have. And so really the whole idea of the BioFoundry is built on this one really kind of simple sentence. Glycans are simply under-taught in the classroom and they are understudied at the research bench. And that's a fact, right? And so the question is, how do we get up into the central dogma, right? Everybody probably, right, who graduated high school and beyond knows that DNA encodes RNA and RNA encodes protein, right? The central dogma of molecular biology. But that doesn't address glycans and lipids. So what we really want to do here is try to address glycans and try to figure out how, by building better tools for people, for providing training, providing educational resources, can we improve and get glycans kind of thought about in the same category when people think about nucleic acids and proteins, right? So how do we get there? And so this is kind of our scientific vision, right? So, right, for the last 60 years, kind of in that central dogma, right? You've had genomes to proteomes, right? And what were the big discoveries that drove that, right? Things like restriction enzymes, the ability to do nucleic acid sequencing. More recently, right, genome editing, CRISPR-Cas craze, right? And all of this fed into this central dogma of molecular biology, right, that was integrated also into education and training, right? And, you know, this is a process that's been going on for 60 years at least. And so what we're going to try to do is accelerate our process using things like AI and machine learning that Karen will talk to you about, where we kind of can go from the glycome to the glycoproteome, focusing on glycosyl transferases that Kelly will talk to you about, glycomics that I'll talk to you a little bit about as well as Paris too, and then glycome editing, right, which we won't really get into today, but we have that as well. And then importantly, integrating glycoscience into life science education and training. How do we go about doing that, right? So we've got outreach programs that we're probably not going to talk very much about today, where in the summers, we're teaching high school teachers and helping them develop a lesson plan to do a 1 hour lecture and a 2 hour lab that they can take back to AP biology or AP chemistry classes, right? Because we can't touch that many students, but if we can get 50 teachers, right, each teacher is gonna see 100 students, right? And so it allows for this amplification, right? And so we're hoping to pull this off in 6 to 12 years by using things like AI and machine learning and right, collaborating with people. And so we've got 3 phases. I'm not gonna walk you through it, but right, it's just like building the building, right? You build a foundation, you engage users, you have a few of them, and then hopefully it takes on a life of its own, right? And then you have this integrated resources. And so we're just in this phase 1 plan. And one of the most important things about the phase 1 plan is letting people know what the BioFoundry is trying to accomplish and what we can do to help your research or help your education plans. And so the idea is at the end of these 6 years, right, we'll have impacted the economy, the workforce, research, and education, right? And that's the whole point is, right, to translate our vision, right, and have people buy into it. All right, that's enough of the salesman preaching, right? So let's talk about what we're really, right, what are we really doing? And so this is kind of the way it's broken up, right? We have education and instruction, we have technology development, we have research. We'll talk to you a lot about these 3 today. We also have a lot of corporate partnerships, right? Right, we have a partnership going on right now with a major mass spec company, looking at an instrument that's not commercially available yet, right? And we're going to run it through its paces, right? And so working with startup companies, but also established companies, national labs, et cetera, et cetera. So I'm going to talk to you a little bit about glycoproteomics and glycomics, mass spectrometry, and how we can help you. And so we have some dedicated state-of-the-art analytical equipment that just got purchased in the last 6 months. It's all been installed now, right? So we've got the ability to do ion mobility and multi-imaging, right? So we can image tissues, right? We can also do glycomics. Of course, we also have kind of our bread and butter instruments like the Ascend, which allows us to do specialized types of fragmentation that you need to do if you want to look at intact glycoproteins, right? And so it also gives us the ability to do quantitative proteomics and glycoproteomics. And then the idea is we want to have a lot of samples, right? We want to be collaborating with a lot of people. And so one of the things we've done is we've got a robot now so we can do automation. So the digest is the protease digest, quantitative TMT labeling, right? That's all automated, right? And we don't have to put our hands on those samples, right? And so that allows us to do a lot of samples, right? High throughput. Both of these instruments, right, are capable of running 24/7, 365, right? So just a couple of examples of what we can do, right? So next gen analytics, right? So at the CCRC, selective exoenzymatic labeling is a technology that's developed, and we're using that and offering that as a service, right, for cell surface glycoproteomics. So if you have a cell and you want to know what's on the surface of that cell, and you can do it in a quantitative manner, right? So it could be in this tumor tissue versus adjacent tissue, what's on the cell surface differentially, right? And we can do that targeting in-link glycoproteins, which is kind of what's shown here, or we can do it targeting O-link glycoproteins, right? And so we can talk to you about what's different on the cell surface, right? So if you're looking for targets for your cell type of interest, it either defines that cell type, or, right, that you're going to use to develop a monoclonal antibody therapy, right? We can do that. And we can also help you talk about the glycoform, right? PSA, right? One of the first biomarkers ever that would never get approved today, by the way, right? It's not good enough. But there's actually glycoforms of PSA that are amazingly good markers of prostate cancer, right? And so combining glyco and the protein on the cell surface to get even more selective, better markers, right, is one of the goals. Another one is just doing glycomics, right? And so the way glycomics is currently done is what we call uninformed fragmentation, or just random fragmentation. And this is just an example actually from an embryonic stem cell looking at some O-glycans. And there's all these fragments, right, that we eventually can kind of figure out what's going on. But most of our fragments are not very useful right? At this given mass, there's 4 possible glycans, and the major fragment can be generated by all 4 of those glycans. So, right, it's taking up all my instrument time looking at fragments that I don't really care, they're not diagnostic. And so what we've done is developed intelligent fragmentation, right? Trying to go from kind of singular sequencing to next-gen sequencing, right? Where we do simple patterns in fragment peaks, right? And so we know what these 4 possibilities are. And so we can build a fragmentation tree where we only see a single peak only if that glycan is actually there, right? And we're building these out for O-links and N-link glycans, starting with mammals, which is where a lot of us have our headspace. But the idea is to spread this to all species, right? We want to be agnostic about species. The other thing we can do is glycoproteomics, right? And so for those of you who aren't glyco people, right, unlike most other post-translation modifications, site mapping is just the beginning, right? If you're doing phosphorylation, site mapping, you're done, right? That's my site of phosphorylation. Well, once I know my site of glycosylation, then I run into this concept called microheterogeneity. And so this is a peptide, glycopeptide, asparagine 190, that contains asparagine 192 from a protein called GLA. GLA is also known as Fabrazyme, right? So this is an FDA-approved biologic, and this is the heterogeneity in that biologic at one site. And it's really these 2 forms that are what the company want, right? This is mannose-6-phosphate to help it get into the cell, but it actually has all of these other glycans on that site too, right? And so we can completely define the microheterogeneity of a purified glycoprotein, right, or a biologic. And what we're hoping to do with Cannon's help is also learn just how to predict what the microheterogeneity is going to look like ahead of time, right? Because we see certain proteins get stuck in certain places, and the question is, can we figure out how to predict? And then, right, just another example of using both glycomics and glycoproteomics allows you to see the same microheterogeneity, in this case, the SARS-CoV-2 spike protein. It kind of ruined a couple of years of everyone's lives, right? And right, it allows us to also do modeling where we can take all these glycoforms, put it onto a protein, and we can see, right, in this case, this is working with Rob Woods' group, we can see areas that are actually available for something like a monoclonal antibody to bind, right? So if you're trying to make a vaccine using peptides, and then other regions that are completely shielded by the glycan, right? And so that's just a little bit of an introduction to the glycoanalytics. You'll hear a little bit more about that from Parashar when she talks about the user facility. But now we're going to shift gears, talk about the glycoenzyme resources from Kelly Norman.
Thank you, Lance. Okay, so as a part of the overall organizational chart for the BioFoundry, I'm going to be focusing largely on components of the research and some of its carryover into the technology development that we're going to be doing in the program. And in particular, we're taking advantage of collaborations and studies that are between Cannon's group and our group, and also in terms of Lance's groups, in terms of the glycoanalytic methods, but leveraging AI and kind of protein and enzymology in terms of the glycoenzyme space. And in particular, as Lance mentioned, the key element of this program is to have it be interactive with a user base that it works in a variety of species and has interests and needs in terms of wanting to have certain aspects of their biological system studied in regard to the glycoscience. So it's an interaction between a user base and a research component. And in the case of the research component, we want to be able to leverage machine learning and take predictions deriving from the AI approaches and be able to validate those in terms of either identifying novel functions as GTs or potentially engineering those enzymes, or as Lance mentioned, studying aspects of how you generate uniquely modified glycan structures on individual sites on glycoproteins, and of course leveraging the high throughput glycoanalytic approaches that he's developing. And so aspects of that includes not only AI and machine learning, but also protein expression and being able to then characterize the resulting products from that. And so the overall goals within the research program had a variety of build, test, refine cycles in which we're going to be predicting and validating GT functions across all domains of life in terms of identifying the diversity of glycoenzymes that are in different organisms, and then being able to hopefully predict what their specificities are, and then be able to test that by expression and validation, and then have iterative cycles of being able to leverage that to understand more broadly glycoenzyme functions in a variety of species sources. In addition, we want to then be able to leverage that information for being able to engineer novel specificities, capabilities of defining the rules for donor and acceptor recognition, and being able to modify those in a defined manner to be able to generate new and novel synthetic capabilities. And of course, that is going to be an iterative process that requires build, test, refine cycles also. And then, as Lance mentioned, there's certainly a high importance for understanding how you generate unique glycan structures on individual sites. Even on the same glycoprotein, it's very common for mature glycosylated proteins to have multiple glycan sites, and for the structures and the ensembles of microheterogeneity on each site to be quite different. And what is it about the sterics and the proximal aspects of protein structure that influence its ability to be processed from a precursor to a mature state? And so we want to be able to leverage both glycoanalytics to identify what those structures are, and then be able to hopefully identify rules of how you would then be able to predict what structures are going to be generated on individual sites. So to do this, of course, we're going to be leveraging machine learning, and Cannon will be expanding on this considerably in his presentation. But the idea is to identify the broader set of glycoenzymes and try to identify the unique structural features of those that define donor and acceptor specificity using large language models and other approaches. We will then be working with him to be able to test those aspects of specificity. And in order to do that, we need to have developed robust expression strategies for being able to produce enzymes of diverse synthetic capabilities, and hopefully be able to establish workflows for characterization based on prioritizing the enzymes that would be of most value to the scientific community, and partly being driven by those requests from the broader scientific community. And of course, to do this, we need to have robust methods for construct design, engineering, expression, and leverage platforms for expression that some of which we've developed in the lab previously. In particular, over the last decades, I'm sure many of you know that our lab has developed a mammalian expression platform for being able to produce secreted forms of enzymes of various glycosyltransferase and hydrolase and glycan modifying specificities, and be able to purify those and characterize them. And so we've developed a library of, well, now it's well over 300 or so constructs, coding regions spanning the human glycoenzyme space, and have developed a growing library of glycoenzymes in the plant space. And we want to be able to expand those across the tree of life. And again, the idea is to take what are normally membrane-anchored forms of enzymes, generated truncated forms, and then append our standard cassettes of fusion protein sequences, and be able to purify those and characterize them in terms of enzyme specificity. In general, at least the eukaryotic version of those enzymes tend to be very much a challenge to produce in non-mammalian systems or non-eukaryotic systems. But we are trying to leverage the expanding capabilities of bacterial expression, in particular with a relatively new postdoc, Hirakan, in my lab, and a growing group of undergrads that are helping in this process, trying to test a matrix of fusion tags, host strains, strategies for bacterial expression, taking advantage of some of the newer advancements in bacterial expression for potentially being able to provide capabilities for chaperones and disulfide bonds and other approaches, including engineering of the protein coding sequences to hopefully be able to jumpstart some of those proteins for expression in non-mammalian systems. But we also want to be able to leverage the capability of characterizing that and taking advantage of AI approaches to do that. And we anticipate that we will be able to have AI-driven prediction of specificities and curate and prioritize GTs for being able to then test the hypotheses. And of course, the question is, can AI tools reasonably be able to predict donor and acceptor specificity? The problem is in protein structural databases, there are a limited number of empirical examples of glycoenzymes with bound substrates. And can we leverage protein modeling tools and provide the basis for a hypothesis for donor and acceptor specificity? And then can we test those with a test set of hypothesized donors and acceptors and express and be able to examine what those specificities are? Can we then define active site determinants for what defines those specificities? And can we test that by mutagenesis and biochemical characterization? And can we expand that by engineering unique capabilities for donor and acceptor specificities and alter the specificities of those enzymes and be able to test that experimentally? And so one of the approaches that we have been leveraging, and certainly Cannon's lab has been leveraging too, is the growing AI space for being able to do protein modeling and engineering. And as you are all undoubtedly aware, the development of the AlphaFold modeling approach has been has had an incredible impact in being able to predict protein structures. And the web-based version of it that came out late last year has capabilities of taking a primary sequence, and in fact, it can be multiple chains of primary sequence, be able to append post-translational modifications, and has a limited capability of having ligands and ions being added. Also, just a couple months ago, there's an open source version of AlphaFold 3 that has expanded capabilities, in particular in regard to ligands and the types of components that you can model associated with those proteins. But the problem is that the formats of the input of capabilities of those ligands or post-translational modifications are quite limited at the present time. And so the question is, given those limitations, can you be able to predict taking primary sequence and a collection of computationally generated ligands and ions and be able to predict bound conformations so you could leverage that in terms of understanding substrate interactions? An example that a grad student in my lab, Daniel Tirani, has been working on over the last couple of months is a complex system that is a wonderful test case for this type of modeling and interaction. It's a complex that's involved in chondroitin synthesis, and it's a heterocomplex of two different proteins that form an obligate heterocomplex for catalytic activity. Based on biochemical and mutagenesis studies, we know now that these two domains of this protein are the active domains in making the heteropolymer of the chondroitin backbone. But these two, and based on a simple AlphaFold modeling, we know that these two in this heterodimer model are the positions of those active sites. By comparison to these two domains, which are inactive and act as a scaffold and a chaperoning type of arrangement to assemble the synthetic component of the heterocomplex. And so the question is, based on the open source AlphaFold 3 and the two protein sequences and inputs of computational files of donors or ions or a polymer of a given glycan that would be an acceptor for that protein complex, can you predict the positions of binding and be able to predict how those substrate interactions can occur? And the answer is yes. You can just do an AlphaFold 3 prediction and be able to identify the locations in the heterocomplex in the CHSY3, but none in CHPF. And you can then look at the actual bound conformations of the donor and the acceptor. And in fact, it will predict a position of the catalytic base and appropriately find the hydroxyl and the position of the donor to basically model a Michaelis complex. I have to move faster then. For both of these sites, and effectively gives you a sense of exactly how substrates and catalysis would occur. You also can do these not only with a free glycan, but you can do this with a glycoprotein substrate. And Hari in the lab has been able to model a complex of a glycoprotein and ask the question, does it bind? Can you model the binding of the glycan component into the active site of an enzyme that would normally process that glycan? And this was a modeling of EPO with a glycan structure on it, and the active site of an enzyme that adds a core fucose of the glycan. And you can see the bound complex, and it actually matches exactly what our crystal structure of that protein complex is, only in this case using a glycoprotein acceptor. So you're able to then append other glycan structures in other contexts and ask the question of whether you can then get information in regard to the capability of these enzymes to modify various sites. So as I mentioned, one of the biggest problems is there are challenges in glycans. They're poorly represented in alpha fold, and the input oftentimes doesn't capture that specificity. We were just lucky in the chondroitin synthases and the FUT8 that it worked. But we've had many examples in which it doesn't work effectively. But fortunately, in just the last couple of days, Chin in the lab has been able to identify a universal syntax for being able to model the glycan structures with a relatively simple coding for AlphaFold model. It appears to work for a diversity of linear and branch structures, and it should work with all known monosaccharides, even some of those unusual plant and bacterial monosaccharides, including all either as ligands or post-translational modifications. And it'll open up all sorts of glycoscience. For example, glycans binding as substrates, what fits in the active site and what doesn't. Substrates is PTM. What are the steric constraints that influence access to the enzyme sites? Glycans is ligands for lectins or co-receptor functions. Glycans in enzyme protein engineering, or modeling in complex environments of glycans in glycoprotein contexts, virtually all biological systems in which glycans are involved. And I believe it's going to be a real revolutionary tool in understanding how glycans are playing roles at a structural level in terms of biology. It also will leverage a whole host of engineering approaches of the type that have been developed previously in terms of what is termed bump hole engineering, adding additional orthogonal chemistry onto sugars that you can then be able to install onto glycan structures of the type that led to a Nobel Prize by Pirel and Bertozzi in the last year. And many of those workhorse enzymes are actually pretty lousy and promiscuous in terms of the specificities that were installed to be able to carry out those bioorthogonal chemistries. And we'd like to be able to create more specific and directed mutations to drive that. And so we have not only are we developing those technologies in the research component, we also want to be able to leverage those for putting them into technologies that will be distributable to the broader scientific community. Our contributions of this will largely be in generating libraries of enzymes. And the idea is that we want to be able to generate community resources, expression construct libraries, validated enzymes across the tree of life, analogous to our human constructs libraries, and be able to produce those in mammalian and bacterial hosts and be able to distribute them with a resource that we've worked with before in terms of constructs development, and then be able to have engineered components that we will also add, that we would provide to the scientific community. So just like Lance said, we have a collection of year one goals that we'd have both on the research and the technology component, but I won't go through them in detail. These are the types of achievements that we'd like to see on the year one goals, but of course have longer term goals in terms of research and technology distribution too. So I'll hand this off to Cannon now to talk about the machine learning part of it.
Thanks, Kelly.
Oh,
no, you're perfect. You're perfect.
All right, thank you. Kelly, thanks so much. I think I can take questions because he pretty much covered most of what I was going to say with the AI technology, but I will try and So I'll give you an overview because, well, again, first of all, thank you all for coming. I mean, it's wonderful to see all of you here and to kind of share your afternoon with us and to see what we have to offer as part of this really exciting project. So as Kelly pointed out, I mean, the technology aspect of this, and I'm going to be co-leading this, and it's got multiple components. And as Kelly pointed out, obviously developing some of the AI tools for glycoenzyme classification and function prediction. We've primarily been doing this, using more traditional informatic approaches, but now we have to get more efficient, right? So we have to leverage some of these more advanced AI techniques. And so that's one of the major goals. And obviously, Kelly pointed out the technologies for glycoenzyme expression. This will be the work that we'll do mostly in collaboration with Kelly. And as Lance pointed out, we're going to be working with him on essentially developing new technologies for glycoanalytics and developing new models for predicting heterogeneity. The reason why we got pulled into this project was sort of independently, we were leveraging protein language models for protein classification with a focus on kinases. And so when Lance contacted me, he said, well, we'd really like to see, how these technologies could be applied for glycoscience. Now, obviously, I thought that was a really cool idea. And so for those of you who are not used to using protein language models, here's a quick introduction to how it works. So most of the technology, the techniques are borrowed from advancements in natural language processing. So ChatGPT, for example, uses the transformer architecture to really understand the grammar of the English language, right? So for example, if you have the sentence here, an English sentence, the model, when it's trained on a large corpus of text, data, really understands the grammar. And so you can use this contextual information for now downstream analysis, such as sentiment analysis, topic analysis, language conversion, and so on and so forth. So there is also a major effort in using these techniques that have been used for text analysis, natural text analysis for proteins, because proteins are nothing but cell's own language. And so essentially, if you just train a model on all protein sequence data in the UniProt, you can learn the underlying grammar of protein sequences. So that's the idea. And so, we and others have shown that, these models that are primarily trained on protein sequences understand the grammar of how a protein would fold into a substructure, where the modification sites would be, what the stability of a given protein would be, what the cellular localization would be. So those sort of properties, if you will, protein properties, can now be predicted with these models trained on protein language. So we got really excited and thought, okay, now we could begin to use some of these technologies for addressing some of the challenges in glycoscience. And so some of the specifics that we proposed to do, and obviously things have evolved and changed since we proposed some of this, was to essentially fine-tune some of these evolutionary scale models for predicting where the functional sites in glycoenzymes would be, without having to do an alignment, because that's been the most time-consuming aspect for us, doing traditional informatic approaches in glycoenzymes, because these are highly variable proteins, especially the acceptor loop regions are evolving pretty rapidly. And so it's very difficult to use traditional approaches to study those highly variable regions. So now with these embedding-based approaches, we can begin to address some of those challenges. How do we use these embeddings to come up with classifications that capture both sequence variation and structural and functional variation? And how do we use these models for predicting what the donors and acceptor specificities would be? So those are some of the things we've proposed. But obviously, with the AI field, things are moving so fast that things have changed quite dramatically since we proposed some of the initial approaches. So, but we have in the past 6 months been successful in installing this a new server that's going to be dedicated for AI and model training and development. And so this is currently housed in GACRAC, and we're really excited to get this resource here. And we're happy to work with folks who are interested in training new models. This will also be available to them. But in the past 2 months, since we've installed this server, we've been primarily using, it's been working full time, essentially 24 hours and majority of the resource is now dedicated for training new models. But as Kelly pointed out, just when we installed the server, AlphaFold 3 came out. And so we had to essentially, refocus and reshift to seeing, to what extent AlphaFold 3 can perform some of the tasks that we had originally proposed to do. And so obviously the AlphaFold 3 is using a really novel architecture. It's using a diffusion architecture for modeling proteins. And AlphaFold 2 did not use that. And so with this diffusion model, what you're essentially trying to do is to essentially introduce noise in the protein structure, and then you're letting the model learn to denoise and understand the 3D structure, right? So when you do this on a lot of biomolecules, the model essentially learns to predict the structure of any molecule, irrespective of whether it's a protein or an RNA or a DNA or a glycan, right? So, and that's what makes this architecture, the AlphaFold 3 architecture, so applicable for glycan modeling. And so clearly with AlphaFold 3, we had to now go back and revisit some of the questions. How can we leverage AlphaFold 3 for improving glycan protein interactions? And as Kelly pointed out, how do we use this for design and also answering questions related to heterogeneity? So just like Kelly, we've been doing some benchmarking of AlphaFold 3 independently. So we have to join forces. But one thing we realized, and maybe this is something maybe Kelly could attest to, is AlphaFold 3 is good at getting the enzyme glycan, overall positioning correct. But then what we feel is that it still hasn't learned the glycan stereochemistry properly. So here's an example of what you give an alpha configuration of mannose as input. So you specify that using a SMILE string, and sometimes it models the beta configuration of mannose. And so this is a sort of a recurring theme where we find that AlphaFold 3 really does not truly understand the stereochemistry. So just like Kelly, we're trying to go back and see, how we can, rectify this issue. We've also been in communication with the DeepMind folks, and they've made some suggestions. So, but irrespective of these small sort of issues in modeling stereochemical aspects, it just as Kelly pointed out, it's just doing a phenomenally good job with other cases. So this is an example of an unpublished structure. This was brought to us from Lan, who's just joined the group recently, working collaboratively with me and Bree. And so she had the structure of this polysaccharide synthase and this ternary complex. And she said, this is not published, so let's try this out. And when we modeled this, it just perfectly matched the 3D structure, which is quite exciting because here you have a ternary complex that the model has never seen before, and yet it gets the perfect stereochemistry right. So that was quite encouraging. So since then, we've essentially beta tested this on a lot of different proteins. Here's one in collaboration with Lance Wells. So he wanted to actually model the trisaccharide with POMK. And here, interestingly, if you have the trisaccharide linked to the peptide moiety in dystroglycan, it gets the anomeric configuration correct, right? So when you have this other sort of fluorophore or this tag, which was solved in the crystal structure form, it does not get it right. But when you have the peptide linkage, it gets the anomeric configuration correctly. So, but this is all truly exciting because, particularly for me, this POMK is super cool because part of my lab works on kinases and pseudokinases. And for a long time, we thought that POMK is a pseudokinase, because it cannot catalyze phosphorylation of protein substrates. But now we know that it can phosphorylate sugars. And so there are lots of kinases and, that really don't look like a protein kinase. So are they phosphorylating sugars or other substrates? Those are questions we can now begin to answer with AlphaFold and some of the new tools that we're developing. So really finding synergies and bringing in fields, disparate fields, is one thing that I think the BioFoundry should probably explore. And in this case, I think pseudokinase researchers will be really excited to see some of this because they don't think about glycans. They think about proteins, but not about glycans. So that's one of the exciting aspects of, I think, the BioFoundry. All right, so last slide. As Lance pointed out, we're really involved in training. So if you're interested in a project that involves AI ML techniques, we're very happy to work with you. You can either bring in your data sets. We'll work with you to show, how to work with your data, how to do some of the standard machine learning approaches, and sort of point you to the right models to use, because that's not always trivial. So these are some of the things that we're working on, and hopefully in a few months' time have a good set of tutorials and workflows for the broader community. So with that, I want to stop and sort of introduce the team. Well, maybe not introduce, but these are the folks that are really doing the work. And if you have any questions, either contact me or any of the folks here. So it's really a team of computer scientists and chemists working together. So thank you. Oh, thank you. Yes.
All right, Aristu is going to introduce us to everybody to the user facility. For those of you, I've noticed some people are taking pictures, which is totally fine. It's also being recorded, the whole talks. And so we'll put that up on our website just to make your life a little easier.
Good afternoon. My name is Paris Rezadi. I'm the director of the user facility for BioGrade. In addition, being director of overall service at the CCRC. So up to now, everybody's told you kind of what BioGrade is going to do for you. I'm going to tell you a little bit of what BioGrade is going to do for you, but I'm also going to ask you what you can do for BioGrade. So I'm going to tell you what the organization of BioGrade is in relationship to the different components and how we're going to operate. So you've seen this picture before. The user facility is really in the heart of the BioGrade. And what do I mean by user facility? It means every instrument that you saw, everything that we do kind of goes through the user facility. So BioGrade has purchased a number of instruments. They could be in different labs, but they're all part of this user facility. What does this user facility do? Its function is, first of all, to bring in users. So users can be, you guys are potential users for the user facility, but more importantly for BioGrade is users outside the CCRC and users outside the UGA. So we want to interact with people from the whole scientific and the Glycoscience community. So user facility is involved with the research component, is involved with technology development, in education and instructions, and really forming partnerships with nonprofit organizations like whether it's FDA, US Pharmacopoeia, government agencies like USDA, and industrial partners. So basically, the aim is to get as many projects as possible now that we're starting. We have our instruments now in place. We're getting trained on our instruments, is really to be able to get in lots of projects from you guys and from outside UGA. We want to make scientific contributions, and the goal is to publish as many joint publications as possible. So the different components that we talked about was the glycoanalytics, that the lead is Lance, glycoenzymes, Kelly, and bioinformatics. So how do these actually work? What happens with the project? So you may have a project, and I'm going to tell you a little bit more about the glycoanalytics. You may have a project that involves cancer cells versus normal patients, normal people who don't have the disease. Or you may have some kind of, if it's microbial, you may have wild types versus mutants. If it's plants, also you may have isolated your plants from different species or different parts of the plant. What is different about BioGrade is we are not only working with mammalian systems. It's a bigger umbrella, all types of glycoproteins from all different sources. So we want to engage you and the scientific community who have potential projects that are bacterial, microbial, they're plant, they're algal, they're from worms, they're from any source, because we want to expand the glycoenzymes to those fields. We want to expand glycoanalytics, and we want to develop bioinformatics and AI tools for that. So as Lance said, we can do glycomics and glycoproteomics now, but we have a problem because we don't do that well that fast. We can do 2, 3 samples. Maybe it takes a couple of weeks. Sometimes if they're complicated, they're from cells or tissues, it takes a couple of months. And that's not really high throughput. We want to be able to do 50 samples, 100 samples in a couple of weeks. Right now, we cannot do that. If we want to get into the field of really interacting with industry, we have to do high throughput. So we want to get into few thousand samples with the technologies that we're going to develop. So how does it all work? The user facility is here, and the users basically contact us. You may contact me, or outside users, other universities and research institutes and industries contact us, and we initiate projects. They could be glycoanalytics, the enzyme work, or the AI. We also have an internal committee that decides on which projects we actually accept, and they make decisions on how we go about doing the projects, how much time we're going to spend on the projects, what resources we're going to spend on the projects. I'll tell you a little bit about this internal committee later. Then in years 2, 3, or 4, we're going to also form a user committee. This user committee could be made of you guys and also external users that you're going to get together and tell us how we can improve on our, maybe the way we do the projects, which kind of projects we should take on, and maybe guide us on what kind of training we should do. So your feedback, basically, if you want to be part of this user committee, is important to us. So basically, samples can go both ways. We can receive samples from users. We can also give out samples like glycoenzymes and other resources to users. So how does actually starting a project, initiating a project with us? Let's say you have a glycoprotein, or you have some samples, and you want to engage and find out what can BioGrade do for you. So we have 2 types of projects. One are service projects. These are more short-term projects that you contact me, or you may contact one of the co-PIs here. If it's an AI project, you may contact Canon. If it's a glycoenzymes, you can contact Kelly. Or if it's a high throughput glycoanalytics, you can talk to Lance. But eventually, it will all come to the user facility. So eventually, you have to deal with them. So we basically set up a call, or you can come downstairs and see me. And together with Christian, we will come up with a protocol to write a standard kind of a protocol for you, how we're going to do things, a statement of work. And basically, we write a quotation of how much we think this is going to cost BioGrade. Basically, what we do for you is pretty much free of charge. So BioGrade is going to take care of most of the costs. Unless it's really beyond the scope, then we may charge some things for some special reagents or something like that. But the instruments are here, so you don't need to pay any instrument time or instrument upkeep right at the beginning. We may later on start charging more, but right now we just want to get as many projects as in. So, and we have the funds to initiate some projects. So it won't be any cost to you right at the beginning. So there will be an online form that once everything is sorted out with our website, I'll send you an online form that you will fill in, and we can initiate the project. Most of our projects are actually not going to be service projects. They're going to be proposal projects. That's an NSF kind of rule. They will require these more long-term projects, because we don't want a project that comes in and it gets done in 2 weeks and it goes out. We want to develop technologies. The whole point is research, technology, and the user facility. So we want problems, challenging problems that you have, and you need our help to solve these problems. We want something that is going to really extend glycoscience information and technologies. So we really want some challenging projects that are happening in your labs. These are longer term projects. Again, we set up a call, and together we decide with the co-PIs that have the expertise in that area, what are the kind of experiments we're going to do, what are the protocols we're going to develop. You fill in an online form, but together we have to come up with a 2-page proposal. That's again an NSF requirement that needs to be written, and it needs to go for an external review. We have a pool of external reviewers that we've contacted, and they've agreed to really look through our proposals. So we'll send this 2-page proposal. It's not a difficult proposal, and we have experience in doing that, so we can help you put this proposal together. But you know the questions that you're going to ask, so we're going to ask you to initiate that proposal, and we can tell you what kind of technologies we can lend to that project. So together we send this proposal out for review, and then hopefully soon we'll get a review back and we can initiate the project. So what is the, how do we go about this internal review? We're going to have a small committee, so it won't get too complicated. There are lots of committees here, basically. I'll be the chair of the committee, and Christian will join with a rotating PI. Right now, it's Kelly. Rotating key personnel, that's Ian, and the managing director or Lance will join this internal committee to go over the projects and decide the resources we need and how long each project will probably take, and whether the scope aligns with the BioGrade basically aims of the project. So eventually I want to come into what can you do for BioGrade. Basically, we want to engage you as scientists at CMM and CCRC to help us get new projects. So not only your projects are important, but NSF really grades us on having external projects coming in. So we need you to talk to your collaborators, to your colleagues and friends, and tell them about BioGrade. How can you do that? You can talk to your colleagues, of course. You can bring us projects and we can tackle them using our proposed technologies. We'll give you a slide. We'll produce a slide. Lance and I are going to work on this slide that we're going to give you, and we ask you to show this slide when you go into presentations and conferences, to tell just 1 minute about BioGrade and say, these are the resources they have at BioGrade if you want to engage with them. And then suggest online and in-person events to promote BioGrade. So if you have any ideas how we can promote BioGrade in your particular area of research, let us know, because we want to be present at those meetings. There may be meetings that we're not attending to, but you are going and you think BioGrade can be helpful, so let us know. That's all I have, and I'll be happy later on to take some questions.
Great. Thanks, Kirsten. So next up is Aaron Dolan, who's going to talk to us about the education component of the BioFoundry. And while she starts talking, I'm going to download her slides real quick.
Good afternoon, folks. How's everyone doing? You're like awesome. It's last day before spring break, Friday afternoon. You almost made it. I let my students out of class a little bit early today. They were really sad. Super sad. That was a cheer you heard around 10 o'clock this morning, with my students being excited that they were. I'm actually coming from the Active Learning Summit, which is happening right now, over across campus. And so I missed the earlier remarks, but presumably you learned a lot about the interests of BioF in building capacity for Glycoscience research, right? So to do that, we need to build capacity in education, right? How many people took a Glycoscience course as an undergrad? Look at that. Okay, so one. How many people took the Glycoscience course as a grad student? Okay, a few more, but still, look how many people do not have their hands up. So a lot of work needs to be done in that space, okay? So what we're trying to do with BioF Great is use an evidence-based approach in all aspects of our education development and capacity building. And I'm going to walk you through sort of how we think through this as it from a sort of education research perspective. First thing we have to do is actually make people aware there's a thing, actually get them interested. How many people would say you have an interest in glycoscience? Presumably you're here, so that's an indicator of interest. When was the first time you became interested? When was the first time you became interested? Someone willing to share?
35 years ago.
35 years ago, what happened? 35 years ago, that got you interested.
I was on an advisory committee with reviewing an age grant to this center.
You were here?
Not located in this building.
35 years later, you're allowed to talk about it. So you're like, oh, hey, here's an idea. Like, there's a situation that came up, you were in a panel and you learned about an idea and you're like, my interest is triggered. I see a hand here.
16 years ago, I was a grad student struggling to make ends meet, so I picked up consulting work for a pharmaceutical company.
Okay, so I'm working at a pharmaceutical company, that's for you.
And suddenly they're like, oh, we care about glycans on proteins.
You're like, wait a second. Okay, so it's not like it came out of your head, right? You were in the right situation with the right group of people who said, hey, this is the thing. And you're like, no, maybe I'm interested in that thing. It looked like Lance, you were raising your hand, is that right?
Yeah, so I was, grad school.
Yep.
See how many years ago, but.
Yeah. Wow.
And just starting grad school and studying human disease and field work test,
Okay.
right, where they test for certain diseases. And
Yeah.
one of those diseases was a sugar nucleotide disease, right? And
Okay.
I was like, what the
Okay.
heck is a sugar nucleotide disease?
Yeah, but most of it,
Right.
is that the thing? I've never heard of that before. Exactly. And so that got your interest. So this is what's called, Bob, one last thing.
1978.
Yeah, and what happened?
In a lab working on the cell wall of a pathogenic fungus,
So pathogenic fungus, and
completely carbohydrates.
you're like, wait, those carbohydrates. Study those things, yeah.
I learned what's called methylation analysis, where you can see how the monosaccharides are come together. And it made me think that you can get more structures from monosaccharides because they have so many linkages than amino acids or nucleotides.
So many more potential questions, so many more potential functions because of the way carbohydrates are structured. So I love these stories. The challenge is most people in the world don't get these opportunities, right? So we need to think about how do we put out situations that actually will trigger interest. Once interest is triggered, you actually have to have a chance to try it enough to say, is this the right path for me? Is this something that I actually want to continue doing? Once that happens, then an individual will actually say, okay, now I'm going to actually go seek out those opportunities to learn more, do more, contribute more, to the point of having a well-developed interest like so many people in the room have. So BioF Great is going to use this as a foundation for educational materials development. We're going to complement that with two main types of beliefs, fostering two main types of beliefs. First is this idea that I can do it. How many people here are really good runners? Oh. Okay, so people out here are like so runners. How many people hope they never have to run again their entire lives? Yeah, okay. So do you want to go run a marathon? No. You're going to do things that you think you're going to be able to be good at doing. All right. So this is called your expectancies. Can I do it? So we need to make sure that whatever we do educationally makes it clear that folks have the capacity to do a thing. That means we need to not ask first-time runners to start with a marathon. Instead, we start with a little bit, and then we build their confidence, and then they'll do more. And the more you do, what happens when you get better at something? Yeah, you actually want to do it, and then you spend more time doing it, and then you get better at it. It's this positive feedback loop. We also need to make it clear that it's worth it. So I love that our colleague over here said, I work for a pharmaceutical company to make a little bit of additional cash. One way that things are worth it is because of money, but there's lots of things that make things valuable or worth it to us. One is that we actually personally find it interesting. Two is we think it's important. It maybe helps treat disease or helps solve a problem in the world. Three is that it's useful. Like, I'm gonna learn some skills, I'm gonna develop some tools, we just heard about the user facility, this is exactly the kind of thing that a user facility will do, is actually make things useful for people. And then finally, you have to see it as not too costly. So it's not gonna be so hard, so difficult, so I'm gonna have to sacrifice so much that I won't wanna do. So we're gonna use these two big ideas to frame what we do in terms of curriculum and instruction. We're gonna start with little introductory lessons. Introduce the idea, these will be suitable for high school or maybe early undergrads. Then we're already engaging grad students. Who's here creating a video? Anyone of the grad students? Yeah, thank you. That highlight interesting ideas. So again, that situational interest and just enough of a bite of science that people feel like, okay, this is something that I can do. Maybe I want to learn more about it. We also need to make it clear that people can see themselves. So examples of role models that you could see as aspirational. So the way that we do this in education is we actually make it clear, like, here's an example of a person and their actual personal story into becoming the kind of professional that they are today. We call this scientist spotlights. So by telling scientist stories, folks from all different backgrounds and all different experiences can actually see themselves in the work. Then we build on that with maybe an introductory module that's more suitable for an undergraduate biochemistry course, followed by full-on courses, some of which we already teach, some of which we need to develop. And then finally, you just heard about use facilities and how experts can develop their technical skills. So we don't want to reinvent the wheel, though. So we want to first start by identifying and curating existing instructional materials and adapt and adopt where possible. This is where you can help. What have you seen that's good? What do you see that could be good? Then we backward design the material. So we say, all right, what do we want to accomplish? And if we want to accomplish that, what do we need to do? How do we layer that with scenarios, real situations and scenarios that make it interesting? And then how do we incorporate that practice and feedback so that people actually get a chance to get better, see it as something that they can do, and see it as something that is worthwhile. And then finally, we want to evaluate what we're doing. We don't want to just say, oh, I think it's good because the students liked it. We actually want evidence that the students are getting what we want them to get out of it, and then broadly disseminate that. This is the leading carbohydrate research center, I would argue, in the world. Maybe people would disagree with me, but I think so. And so we can be a real resource for the whole community and getting these materials out in a way that really expands capacity. And with that in mind, oh my gosh, I have no idea why I wrote that there. You can tell I was taking, but that should be BioFoundry team, just replace that. And then Megha Tiwari, who just earned her PhD, yay, Dr. Tiwari. And then finally, you. So if you have materials, ideas, interest in any of these approaches, please let me know. And with that, I'm happy to hand it back to Lance.
Thank you so much.